And it's my great pleasure to welcome Professor Gabriela Schlau-Cohen from MIT to give a talk today. A bit of introduction about her first. So she graduated from Brown University with a Bachelor of Science degree. And after that went to Berkeley to do her PhD with Professor Graham Fleming. So Professor Graham Fleming's lab in Berkeley is kind of the pioneer in the field of 2D electronic spectroscopy, and in particular in the application of it to study all sorts of light harvesting photosynthesis process. And that's what Gabriela did for her PhD work. In fact, pioneering using 2D spectroscopy to look at light harvesting complex 2, which is a system that's dear to our research here in NTU as well. From that, she graduated with her PhD, and after that, move on, deciding that 2D electronic spectroscopy is not tough enough for her. She decided to do more a postdoc with Professor Moner from Stanford University. As you guys probably know, Professor Moner is a Nobel Prize winner in single molecule spectroscopy, and that's where Gabriela did her postdoc work for 3 years, I believe. And in 2015, she got a tenure track position at MIT, and she is now doing forefront research work combining single molecular spectroscopy with ultrafast spectroscopy to look at all sorts of interesting phenomenon in exploring the energetic and structural dynamics of interesting biological systems, and that's what we're gonna hear from her. And at MIT, she has won several awards. She has been awarded the NIH National Institute of Health Director's New Innovator Award, the Beckman Young Investigator Award, the Camille Dreyfus Teacher Scholar Award, and so on and so forth. She's currently the Associate Director of CIFAR's Bio-Inspired Solar Energy Program, and on the advisory board of CAMP. And she's also a minor media celebrity, I guess, because she has appeared in Canadian Broadcasting Corporation's radio show, Quotes and Quarks, and also on Newsweek. So today we're gonna hear from her interesting work on combining ultrafast spectroscopy and single molecular spectroscopy on looking at carotenoids in light harvesting complexes. All right, so take it away, Gabriela. Thank you. Thank you for being here.
Thank you, Hao-Seng, for the introduction and also for the invitation. It is nice that despite the challenging circumstances we're all under, to still have the opportunity to talk science and share some of the work that we've been doing in my group. So today I'm going to focus actually on the ultrafast side of our group and specifically talk to you about our work looking at light harvesting complex 2, LHC2, which many of you know quite well. And so when we think about understanding the system, the first thing that we know is that photosynthesis is the powerhouse for almost all life on Earth. So photosynthetic organisms are able to achieve this remarkable feat. But what we want to know is how. How is it that they are able to capture enough energy and use to power so much of life? And so we can think about this process by beginning looking at the solar spectrum. So we know sunlight is a broad light source, and plants absorb across a large swath of this. So what I've overlaid on the solar spectrum, shown here, is the absorption spectrum for light harvesting complex 2, or LHC2, which is the primary antenna protein in green plants. And like I said, it's something that I know is well known to many people in the department. And so if we're gonna think about LHC2, what we know is that this and other antenna proteins capture and collect energy and then use that energy to drive processes like charge separation, water splitting, and all the biochemical reactions of photosynthesis. But what's remarkable is in this process of gathering that energy, it needs to gather and then transfer energy rapidly over an enormous energy gap, over 50 times kT. So one of the questions we're interested in addressing, how is it that energy is able to rapidly and efficiently relax across this large energy gap? Now, another thing that happens is that sometimes, as we all know, it's sunny. And when the light intensity increases, that light energy in excess of what's needed for photosynthesis can generate reactive oxygen species, which cause cellular damage. And so what plants do is they actually activate non-radiative decay pathways that convert that excess energy to heat. So what we're also interested in knowing is how is it that excess energy is dissipated? So these are the two central questions that we're going to address in the work that I'm going to tell you about today. And so in thinking about this work, we can begin by going to the structural model of LHC2. And so here's the structural model from X-ray crystallography. So what we can see is that we have a protein scaffold shown in gray, and densely packed within these protein scaffolds are two types of pigments. The chlorophyll, shown in green, which are the primary light harvesting pigment of photosynthesis, and then the carotenoids, shown in pink. They're an accessory light harvester and they serve some other roles. And so collectively, these pigments give rise to this absorption spectrum. And what I've colored here is the different regions associated with the different pigments. And so if we want to understand these pigments and their interactions, what we need is a probe that maps out the photophysics across the visible region of the solar spectrum. And so one probe that's able to generate that kind of map is two-dimensional electronic spectroscopy. So 2D spectroscopy is well known, like LHC2, to many of you, but fundamentally what 2D spectroscopy is it's a correlation map where it correlates excitation energy and emission energy. And so what that means is in a 2D spectrum, peaks that appear on the diagonal, where excitation and emission are the same, generally correspond to the linear absorption spectrum shown above. Now in 2D spectroscopy, we're also able to measure the correlation between excitation and emission as a function of a delay time between those excitation and emission events. And one of the things that emerges from that is off-diagonal peaks, such as what's shown here, where you absorb at a higher energy, in this case the carotenoids, that leads to emission at a lower energy, in this case chlorophyll, and the appearance and time evolution of these off-diagonal peaks directly reports on carotenoid to chlorophyll, in this case energy transfer. And so if we want to use this technique to probe LHC2 and understand the relationship between the chlorophyll and the carotenoids, what we need is femtosecond time resolution, because that's the time on which these processes occur. We need a broad bandwidth that spans both pigments, and we need a high signal to noise ratio. LHC2 has complex kinetics, and that high SNR is required to disentangle this process. Now, one challenge is that getting these last two criteria, the broad bandwidth and the high SNR at the same time, is challenging because most broadband laser sources fluctuate. And so as a result, a lot of the 2D work has focused on one region of the spectrum. Now, this has been powerful. I've done some of this work. Hao Sang has done a lot of this work that have allowed us to disentangle the pathways within the chlorophyll Q states. But if we want to expand this box to understand the carotenoids, the chlorophyll, and their interactions, what we need to do is expand the bandwidth. So the first thing I want to tell you about today is our work expanding the bandwidth to meet these requirements for 2D spectroscopy. So to begin with, I'm going to tell you a little bit more about how we do 2D spectroscopy. So we do this in a non-collinear geometry where there are two laser pulses incident on the sample that collectively serve as the pump or excitation pulse to excite the system. And then there's a time delay with a probe pulse, and that time delay, labeled as capital T, is the time over which we monitor the dynamics or the evolution of our system. And then that probe leads to a stimulated emission. And so that stimulated emission, we interfere with a reference pulse, spectrally detect those, the signal and the reference pulse, and then detect in the frequency domain, or spectrally disperse and detect in the frequency domain. Now, traditionally what's done is we detect this signal, which arrives at the detector along with scatter from all of these different pulses that are required to generate and detect the signal. And so traditionally, these are detected together, and then the individual beams are blocked slowly after some averaging of the signal or at the end of the experiment. And so that slow detection and subtraction leads to low signal to noise. So we can quantify this, and we did this using a white light source, gas filamentation white light, because that's an easy way to generate a broadband spectrum. And we measured this for Nile blue, which is a laser dye. So we can take our 2D spectrum, take a slice out of the 2D spectrum, and look at it in the time domain. We can benchmark the noise by looking at the region after which the signal has decayed, and then compare it to the signal, and we see that this traditional subtraction approach has a signal to noise ratio of around 10. But what we can do instead is we can take our apparatus, we can add choppers, and a high-speed detector so we can read out every laser shot. This allows us to detect the signal and scatter, and then on the next one, scatter alone, then signal and scatter, and so on and so forth, to subtract on a shot-to-shot basis, which allows us to subtract the scatter much faster than many of the fluctuations in the apparatus. And so what this gives us, if we compare to the traditional approach, is in the shot-to-shot acquisition, we have a signal-to-noise ratio of around 140. So this is a 15-fold improvement in signal-to-noise ratio for this ultra-broadband source. Every ultra-broadband source has its own noise profile, but for gas filamentation and argon, which is a straightforward way to generate white light, we're able to see this improvement with these easy improvements to our setup, the addition of choppers along with high-speed detection. So what this allowed us to do was to look at LHC2, not just in one region of the spectrum, but to generate a broadband spectrum that spans the chlorophyll and the carotenoid region. And so with this technique, we can address those central questions of how is it that energy relaxes across a large gap, and how is it that excess energy is dissipated as heat. So for the remainder of the talk, I'm going to discuss using this approach, this ultra-broadband 2D spectrum on LHC2 to address these questions for green plants. Beginning first with this one, looking at how is it that energy relaxes across a large energy gap, or this process of spanning over 50 kT rapidly and efficiently. So when we think about this process, we can think back to our molecular structure. And instead of looking at the absorption spectrum, we can think about it in a simplified picture of the electronic structure, where we have the carotenoid excited states here, and then the chlorophyll excited states here are the lowest energy ones, which are the Q states, which includes QX and QY. Now, if we excite in the blue region of the spectrum, the carotenoid, that leads to this rapid energy transfer to the chlorophyll, as I mentioned, and then it's the chlorophyll QY that gathers the energy that's used to drive the reactions of photosynthesis. And so understanding this process is challenging, in large part because of the nature of the electronic structure of the carotenoids. So it's well established that the carotenoids have a bright S2 state and then a dark S1 state. But there's also hypotheses that there are additional dark states along with the S1. And so the exact number of dark states, as well as their energetics, is difficult to identify because they can't be directly interrogated because they're dark or optically forbidden. So there are open questions about the number and energies of these states. Now, another challenge is that the electronic structure of the carotenoids is variable. So if we look at these carotenoids, they're long conjugated polyenes. Their conjugated electron density as well as their structure is very sensitive to the local environment. And this is illustrated by these two luteins. These are the two central carotenoids of LHC2. They're found in the center. They're chemically identical. Refer to them as lutein 1 and lutein 2. But what's different is because they're in different protein pockets, that leads to a shift in the transition energy of 700 wave numbers over 3 kT between them, just by their different environments, despite their chemical identity. So we want to understand, how is it that this excited state manifold is facilitating relaxation, and whether these differences in electronic structure that we see appearing in the carotenoids have a functional role. And so to investigate those specific questions, we look at this ultra-broadband 2D spectrum, which spans the chlorophyll and carotenoid region. And so we want to know, first, what are the energy transfer pathways within the higher lying states, these pathways that have been inaccessible before. So to address that, we zoom in on this region that has the chlorophyll-Soret bands as well as the intra or intercarotenoid dynamics. And so this is a zoom out of that region that I just had in a box. And what we can see, if we look at these two boxes, this is where we excite the lutein 1 and the chlorophyll-Soret and measure population onto lutein 2. We see a rise of these two cross peaks, which tell us that energy is transferring from both the chlorophyll and lutein 1 onto lutein 2. And we also see that this is happening really quickly. So we see that energy is transferring among these higher lying states onto the S2 state of lutein 2 in less than 100 femtoseconds. And so if we're thinking about the central question of how energy is relaxing across a large energy gap, we see that energy from higher lying states is rapidly collecting onto the lutein 2 S2 state on this sub 100 femtosecond timescale. So next we can go back to our ultra broadband spectrum, and we want to know, can we disentangle the energy transfer pathways from those higher lying states down to the chlorophyll Q states? And to do that, we look in this region indicated in green, which gives us a direct readout of the carotenoid to chlorophyll energy transfer, because this is exciting into the carotenoid and then probing the emission of the chlorophyll. And so what we see in this region, this is again a zoom in of that region of the 2D spectrum, is that we see stronger cross peak intensity for lutein 2 than for lutein 1. Now we know that the intensity of the cross peak reflects the amplitude of energy transfer from that carotenoid onto the chlorophyll. And so that larger amplitude of lutein 2 tells us that lutein 2 transfers more to the chlorophyll than lutein 1, or than the other carotenoids. And so again, getting back to the central question of how energy efficiently relaxes across a large energy gap, we see that energy from a higher lying state rapidly collects onto lutein 2, and that lutein 2 primarily transfers energy to the chlorophyll. So both of these observations, or this analysis of the 2D spectra, show something about this special role for lutein 2. And so what we wanted to do is look a little bit more directly at lutein 2. And so to do that, we can go to this 2D spectrum and we can map out the photophysics of lutein 2 by looking at this slice of the 2D spectrum here. So our x-axis is our excitation axis again. So this is excitation into lutein 2, and then looking at what happens to our emission frequency. So we take this slice, which is again the excitation into the S2 state of lutein 2, and we stack these slices, not just for one delay time, but for a series of delay times. So this is a stack of these slices where this is the emission frequency now on the x-axis, and on the y-axis is the time delay between excitation and emission. So here we've subtracted the population dynamics, the decays, and what we see is an oscillation. We can fit that oscillation, and we see that at 18,500 wave numbers, we have an amplitude minimum as well as a phase jump. So the amplitude is shown here in black and the phase is shown here in gray. And so this energy, 18,500 wave numbers, is the energy that was previously proposed for an additional dark state referred to as SX, or an additional optically forbidden state. And what we know is that this sort of oscillation and this phase jump and amplitude minimum is a reporter of a wave packet. And that wave packet, the wave packet is on a potential energy surface with a minimum at 8,500. So that's a signature of an energy minimum at a potential energy surface of a lutein SX. So what this is telling us is we've excited into the lutein S2, and we've had some sort of non-adiabatic transition, because that's the only way we could maintain phase coherence after excitation. And we see this to generate a wave packet on the lutein 2 SX surface. And that this non-adiabatic transition occurs in less than 20 femtoseconds, because we see this oscillation begin at 20 femtoseconds, which is the shortest time point that we can extract these kinds of dynamics. So this is a vibrational wave packet, but what this allows us to do is identify this potential energy minimum that allows us to see a dark state that isn't directly accessible using traditional methods. So in going back to our spectrum, what we can now see is there's an additional state on SX, and similar analysis at other frequencies tells us that this SX we only see on lutein 2. So it's exclusively on lutein 2, and it isn't on any of the other carotenoids, even the chemically identical lutein 1. So that tells us that the presence of SX is sensitive to the conformation of the carotenoid or the electrostatics of its surrounding environment in the protein pocket. And this potentially addresses the debate in the literature about the number of dark states, because here, by simultaneously looking at these 4 carotenoids using the same method, we see only one has SX. So it's not that it either exists all the time or doesn't, but instead that it really depends on the nature of the binding pocket for the carotenoid. And in terms of looking at how energy efficiently relaxes across a large energy gap, what we see is that there's this SX state on lutein 2, and what other analysis that I'm not going to show today, but global analysis that the data shows, is that it's observed on lutein 2 and it facilitates carotenoid to chlorophyll energy transfer, where we see that this state can also transfer energy to the chlorophyll QY. So collectively, thinking about this question, we've been able to put together this picture of the photophysical pathways that connect those higher energy or blue states of the chlorophyll Soret and the carotenoid S2 with the lower energy red states of the chlorophyll Q bands. And in this way, we see that plants are able to absorb across the visible region of the solar spectrum, but still rapidly transfer energy down so it can be used for photosynthesis. And this tells us that lutein 2, this one carotenoid, is playing a special role and serving as a nexus for light harvesting in plants. So this is the first part of the applications, looking at how energy relaxes efficiently. Now I'm going to switch gears a little bit to address the second question. How is it that excess energy is dissipated as heat? And so if we want to think about this process, so far we've been focusing on the photophysics of the carotenoid and chlorophyll on basically this half of this Jablonski diagram, thinking about the light harvesting and rapid energy transfer. But what we also know is when there's high solar intensity and there's too much energy collected. There's these dissipation processes, which I mentioned before, where that excess energy is safely converted into heat. And these are the dissipative processes, or the photophysics, that emerge under highlight or sunny conditions. So the photophysical pathways in plants actually switch depending on the weather. So we want to understand also, what are these photophysical pathways? And one key aspect to this is that carotenoids are really good at dissipating energy. They're long conjugated polyenes, so they have a lot of degrees of freedom for rapid internal conversion. They have picosecond lifetimes by which the S1 state returns to the ground state. So it's likely that they play a role in these dissipation pathways. So we want to know, what are these photophysical pathways of dissipation? And what they are is actually widely debated in the literature. So there are two proposals that involve the chlorophyll and carotenoid interactions. One in this region of the spectrum. One is chlorophyll-to-carotenoid energy transfer, where the chlorophyll Q states can transfer to the S1 state of the carotenoid, which can then undergo that rapid internal conversion back down to the ground state. So this is the most straightforward mechanism. It's easy to imagine these pigments that are close together. Energy will transfer from one to the other, and the carotenoid is good at dissipating this energy. But there had been quite a bit of pump probe measurements to look for this process that hadn't been able to see it. So partly because of the inability to observe this process, an additional mechanism was proposed, and that is a delocalized chlorophyll-carotenoid excited state. So in this mechanism, interaction between the chlorophyll and carotenoid gives rise to an electronic structure where the excited states are linear combinations of the two pigments. And so that would lead to the chlorophyll QY states, or at least some of them, having enough carotenoid character for a fast non-radiative decay, which would allow a dissipative pathway to occur. And so the advantage to this is there had been some indirect spectroscopic signatures observed of a delocalized interaction, of an interaction between chlorophyll and carotenoids. It's kind of delocalized state. But the disadvantage of this mechanism is it requires extremely strong coupling and much stronger electronic coupling between the chlorophyll and the carotenoids than you would expect based on our structural models of LHC2 from X-ray crystallography. And so part of the reason that resolving this debate has been so difficult is, again, because they both involve S1. And S1 is a dark state, so it can't be probed directly. There's no oscillator strength. So it can only be indirectly observed, but because there is an optically allowed transition from S1 state to the manifold of higher lying states, Sn. So it's observable via this excited state absorption process. And that's what we're going to use in the experiments that I'm going to tell you about today to look at the photophysics involved with this S1 state. So again, we take our ultrabroadband 2D spectrum. And what we want to know in this case is, can we benchmark the dissipative photophysics of LHC2? And we zoom in on this region of the 2D spectrum here, because this is the region that will allow us to get chlorophyll to carotenoid energy transfer. And so this is the zoom in of this region. And if we look at this square here, this is where we excite the low energy chlorophyll QY, and then we probe that excited state absorption of the carotenoid S1. And so if we look at this, the dynamics associated with this position on our spectrum, what we see here is a rise in the peak intensity with a time scale of around 400 femtoseconds. Now we can also look at this box right here. So this is the excitation into the chlorophyll QY, and then the detection of the chlorophyll QY. And what we see here is a decay on that same time scale of 400 femtoseconds. So this is reassuring because we're extracting very similar timescales from these two separate points on the spectrum. And this correlated decay of the chlorophyll and rise of the carotenoid population after excitation into the chlorophyll indicates directional energy transfer rather than a delocalized excited state. Now, delocalized excited states or strong interactions can be observed in 2D spectroscopy, but they would have an instantaneous rise, not this correlated rise and decay that occur with a time scale that we can measure and extract. So what that tells us is, in thinking about this central question of how is it that excess energy is dissipated, we see that it's dissipative energy transfer that's observed, this dissipative process, instead of this delocalized excited state. Another thing that's notable about this process is that the 400 femtosecond timescale that we had extracted from our measurement is around an order of magnitude faster than the timescale that had been predicted based on predicted using theoretical calculations. So this tells us that those straightforward calculations actually aren't the whole story, and it may be a little bit of a mixture of the two, in that it is a directional energy transfer, but that interaction is stronger than you would expect. And so that may be what had caused those signatures that were observed previously. And so again, getting back to this summary of using 2D spectroscopy to span the chlorophyll and carotenoid regions, if we're looking at this question of how excess energy is dissipated as heat, we can see that dissipative chlorophyll-to-carotenoid energy transfer is observed. So we see that this process is occurring within LHC2. So that allows us to benchmark that this dissipative pathway exists. But in order for a dissipative pathway to be photoprotective, it has to be activated under high light conditions. So it has to be activated by high intensity. So what we want to know is, how is it that this dissipation is or could be activated? And so you can also think about that looking at this picture right here. So what we see is that there are regions of the leaf that have much higher sunlight, that are in direct sun, and then regions like what we see over here that's in the shade. And so then as the shadows move, or as the sun moves, or the wind blows even, there are changes in the solar intensity that each individual region of the leaf experiences. And so for a dissipative pathway to be photoprotective, again, as the sunlight increases, there has to be activation or enhancement of that dissipative process. Now, what's known from previous biochemical measurements is that there are 3 different molecular parameters that activate dissipative measurements. The first is a drop in pH within the photosynthetic apparatus. The second is a change in the organization of the proteins within the photosynthetic membrane. And the third is a change in the chemical composition of one of these carotenoids. So it's been well established that these 3 molecular parameters are what's responsible for activating dissipation. But what isn't really known is how. What is it that they do, and what kind of impact do they have on the photophysics? Now, it's also established that the drop in pH leads to a protonation of a homologous protein called PSBS. And so I'm not going to focus on that in this talk right now, because I'm looking at what can happen to these dissipative pathways in LHC2. And so now I'm going to talk to you about looking at these 2 different molecular parameters, the membrane and carotenoid composition, to think about what role these might be playing in impacting the dissipative photophysics. So beginning first with looking at the membrane. So what we know is that the membrane itself introduces new interactions. So far, I've been talking to you about experiments done on detergent solubilized LHC2. But in vivo, LHC2 is found within a phospholipid bilayer membrane. And so there's real differences in these 2 environments. For example, there's essentially a reversal of forces. In detergent, the hydrophilic head groups interact with the aqueous environment and pull outwards on the protein, whereas in the membrane, there's lateral membrane pressure along the plasma membrane that pushes inwards on different regions of the protein. So we know that the membrane introduces new interactions, but what we want to explore is how those new interactions, or how differences in the environment fundamentally impact the dissipative photophysics, because that's what would give us a knob, again, to activate those dissipative pathways under sunny conditions. And so we want to explore what happens in the membrane. And so what we need is a probe that allows us to explore the interactions with the membrane environment. And so the tool that we use in my group is an emerging biotechnology called nanodiscs. So this is a way in which you can mimic membrane environments in vitro. So what you do is you take your membrane protein, in the experiments I'll talk about, we take LHC2, and you self-assemble around it the lipid bilayer, and then you hold together this protein-containing lipid bilayer disc with a belting protein. So this is an amphiphilic protein that's lipophilic on one side, so it assembles that membrane structure, and it's hydrophilic on the other, so it solubilizes that disc for spectroscopic interrogation. And so we assemble these discs, and to give you a sense of what they look like, here's a TEM image with a few representative discs, as well as a histogram that shows the diameter. Sorry, I realized the axis here got cut off. So the average is 25. This is 30 nanometers. And this is 20 nanometers here. So this shows a histogram of the distribution of individual discs. So this allows us to solubilize LHC2 while introducing some of the interactions that would be occurring within a near-native membrane environment. And so we can then probe the membrane-embedded LHC2 and look at the impact of those new interactions on the dissipative photophysics. And so this is the same 2D spectrum and the same traces that I showed you before that show that dissipative chlorophyll-to-carotenoid energy transfer. And what we see in the membrane environment is an enhancement of this process, both in terms of the rise of the carotenoid population and the decay of the chlorophyll population. So fitting this data shows us that there's a 14% increase in the amplitude of this process within the membrane environment. And so that doesn't tell us exactly how it's activated in vivo, but it does tell us that this dissipative pathway is sensitive to the environment. So any changes to the local environment, such as changes to the protein organization within this membrane, would impact the amplitude of this process. So it tells us that this is potentially a dissipative process that could be activated by some of the changes known to occur under high-light conditions. So now this is benchmarking the dissipative processes, but we also want to know where is this process occurring within the structure of LHC2 and how is it getting impacted. So to think about that, we can go back to a different technique. We can go to another technique, circular dichroism. So circular dichroism is a tool that uses the difference between the absorption of circularly polarized light, and it's a sensitive probe of the spatial organization of your excited states. So we use circular dichroism to investigate LHC2, first in the carotenoid region. And so these are the CD spectra in both detergent and in the membrane. And what we see is there's changes in the CD spectra in two different regions, and those regions are the two peaks associated with the carotenoid lutein 1 and the carotenoid neoxanthin. So that tells us that the structural changes that are occurring in the membrane, which we know enhance the dissipative photophysics, are associated with these two carotenoids. Now we can also look at the chlorophyll region, and what we see is that in the chlorophyll region, there are again changes. They're subtle, but there are changes in two regions, And previous work associated those two regions with the chlorophylls that neighbor lutein-1 and neoxanthin. So again, this is consistent with our measurements of the carotenoid region. So the fact that these are the two regions that change in the membrane tells us that one of these two regions is likely the site responsible for the dissipative photophysics observed. And for neoxanthin, the carotenoids are much further away than they are for, or the carotenoids in the chlorophyll are much further away than they are for the chlorophyll that neighbor lutein-1. So that makes lutein-1 the likely site of the chlorophyll-to-carotenoid energy transfer, or the likely site of this dissipative pathway. So what these two different pieces show us is that for these two chemically identical luteins found in the center of LHC2, that they really have opposing roles. We saw in the first part of the talk that lutein-2 serves as a nexus for light harvesting, having this previously unobserved dark state, SX, that bridges the energy gap to connect the blue region of the solar spectrum with the red region of the solar spectrum for rapid relaxation to collect energy across the solar spectrum. So it plays this critical role in light harvesting. In contrast, we see that lutein-1 is the likely site of this dissipative photophysical pathway, where it is likely the carotenoid that accepts energy from the chlorophyll and then rapidly allows that energy to decay down to the ground state. And we saw that that cluster of pigments responsible for that process is sensitive to the local environment. So we see that the protein pocket is actually able to tune the function of the pigments. And in this case, we see this exquisite tuning of the lutein-1 and the lutein-2 to introduce these dual roles of light harvesting and dissipation. So in thinking about how dissipation is activated, we see that this chlorophyll-to-carotenoid energy transfer can be enhanced by the environment, such as by changes to the protein organization known to occur under high light, and likely that happens on lutein-1. So now the last thing that I want to tell you about today is this third point, looking at the role of this third molecular parameter, carotenoid composition. So this is again that slide that I showed you before, where we're looking at the dissipative energy transfer through the rise of population in S1 and the decay of the chlorophyll in the membrane environment. But now what we also know is within LHC2, one of the carotenoids gets enzymatically converted under high light from violazanthin to zeaxanthin. And so it's known that this process happens under high light conditions, but it's not known what role that plays in the photophysics or mechanistically what that does to activate dissipation. So we measured this dissipative process for with violazanthin bound and with zeaxanthin bound. And what we saw is compared in these two traces here. So this tells us that although we know that the carotenoid in LHC2 changes, it leads to no changes in this dissipative pathway. So that suggests that other parameters must be required to have a full understanding of the role of zeaxanthin in dissipation and photoprotection. So in terms of this third parameter, carotenoid composition, while it's known to change under high light, likely has an indirect role in activating dissipative pathways. So fundamentally, using ultrabroadband 2D spectroscopy, what we've been able to do is not just see this dissipative chlorophyll-to-carotenoid energy transfer that allows excess energy to be converted to heat, but see that that pathway is sensitive to the environment, which provides a knob that could activate it under high light conditions. And we see that it's independent of the composition of the carotenoid, even in the membrane environment. And so collectively, what these results show is that carotenoids in photosynthesis are optimized for these dual and seemingly opposing roles of both light harvesting and photoprotection. And it's really potentially the design of the protein that allows these pigments to serve these different roles. So finally, I want to end by thanking the people that actually did this work. So essentially everything that I told you about today was done by a really talented former graduate student, Minjung Sun, who's now a postdoc at University of Madison. She led everything from the design to the construction of 2D apparatus, all the way through all the LHC2 measurements, as well as a bunch of stuff I didn't have time to talk to you about today. Former postdoc Sandra Mosquera helped with the design and construction of the setup, and a former undergrad, Sam Gordon, did a lot of the biochemistry around the nanodiscs. The work on LHC2 is in really close collaboration with Roberto Bassi at University of Verona and Alberto Pinola at University of Pavia. They provide not just the protein, without which these experiments couldn't be done, but my group is primarily spectroscopists, so a lot of the biological background and insight that helps us interpret and understand the role of these experiments. These are the people that keep the lights on, and I am happy to answer your questions.